So what are we doing? Last law, yeah. No? So we already finished with the first law of law of dominance. We are done. Law of segregation of the end. I gave you one question on law of segregation. No? I gave you one question. We already saw that in the class. Okay. So let us now take the last law, that is the law of independent assortment. Anything that is doubtful till here? Anything that is doubtful? Where's the other girl? Where is she? You were absent the other day. 2 days you were absent. No, only one class you were absent. Okay. Anything that is to be clarified?
Miss, in transpiration factor, Gannon's photometer, we have to write the rate of water intake, not the rate of transpiration.
Rate of transpiration.
Rate of transpiration. Gannon's photometer. We generally don't write that because the Gannon's photometer, if you see the entire device, it has a plant chute, and whatever amount of water the plant chute gives out, almost same amount of water is taken up by that glass tube from the beaker, and that is what is moving. So actually we are not measuring how much of the water is lost, but indirectly we are measuring how much of the water is taken from the beaker by that glass tube, and that is almost equal to what is gone by the plant. So what is this apparatus measuring? You are actually to write the rate of water intake by the plant. So actually we are talking about this plant chute. Now how did we take this? There was a tube like this, no, here, and then we had taken the sugar plant here. And whatever amount of water gone by the way of transpiration, that much of the water is taken. So if I talk about the plant, that much of the water is taken. And that is again shown by the amount of water taken from the beaker by this glass tube. So that was the device, no? So are we actually measuring only here? No, we are not actually measuring here. Related to this, what is happening here? That is what we are measuring. So that's why we should more correctly write down Genome's photometer measures the amount of water intake by the plant.
And also the first chapter in the national following, I think the C and D both are telophase.
C and
D. Like chromosomes become visible as fine long threads. It is telophase. And the D1 is also chromosome lose their distinctiveness and gradually become transformed into chromatic network. That is also telophase. So the national following, which is prophase.
Which is
Prophase.
chromosomes get arranged in order, okay, metaphase. Larger chromosomes move to opposite anaphase. Chromosomes become visible as fine long threads. Chromosomes become visible as fine long threads. This is prophase.
Why?
Chromosomes become visible. From chromatin, they are now chromosomes. From chromatin, which was in telophase, so now they are chromosomes. And then duplication will happen. So now that they are chromosomes, it is always prophase. And chromosomes lose their, this is at the end of the process. Chromosomes are losing their distinctiveness and now they are becoming uncoiled and now they are chromatic. This is telophase.
We have test also.
So that's very good news. You have test.
But then in the second last scheme, chromosome becomes visible as fine long threads. Chromosome as fine long threads means telophase, right? The second last.
Chromosomes become visible as fine long threads. Chromosome becomes visible as fine long threads, meaning they are not talking about chromatin at all. They are saying that chromosomes now that word. Fine long threads, okay, but chromosomes are visible as long threads now. So they are never saying that the chromosomes are visible as uncoiled thin threads. No, if it is uncoiling and from chromosome they are losing their distinctiveness and becoming fine long threads, then it is chromatic. Understood? Bolo.
On genetics and transpiration.
When?
Sunday.
Here. Okay, good, very good it is. You are well prepared with both the chapters? So this is the last topic that we have to discuss here. No, I think sex discrimination already I did earlier, no? So, this is the sex discrimination I already did, no?
Yes.
So this is the last topic. Children, let us complete with this law of independent assortment. Law of independent assortment. Okay, law of independent assortment. What are we going to do here? What is the difference? Here we are going to study dihybrid cross. What was the concept of monohybrid? Only one character. Inheritance of only one character. Now we are to study inheritance of two characters at the same time. Inheritance of two characters at the same time. So if you are studying inheritance of two characters, and that is what is called dihybrid cross. So let us study. We talk about parents. Parent plants. Which are parent plants? Round and yellow seeds. Round and yellow seeds. How do we write down the round and yellowness? Bred with this means breeding happening. Cross. just the you have to write down just the contrasting traits now. When I say round and yellow, which two characters I'm talking about?
The shape and the color.
Shape and the color. So this is one trait. And what is the recessive? Round and wrinkled. Round and wrinkled and green. Wrinkled and green seeds. But how will I write wrinkled and green?
Small.
Okay. Now gametes. Gametes of the parent plant. What is this situation? This situation is something like the cells of this plant has many pairs of chromosomes, and one pair of chromosome has this R, Y, Y. Is this understood? So when the cell divides to show the process of gamete formation, one chromosome will be received by one cell and other chromosome will be received by the other cell. But what are the alleles? RY, RY. How many type of gametes?
2 types. No, 1 type.
How many type of gametes? How many type of gametes you get to see?
1 type of
One
gamete.
type of gamete.
1 type of gamete.
One. Are you sure?
Yes.
Are you sure?
Yes.
Yes.
How many type of gametes? One type. One type. Both the type of cells are having the alleles capital R, capital Y. So one type of gamete. Will it be the same thing that side also?
Oh.
Yes. Why? What is the difference when the two alleles are same for a gene? What is this condition called?
Homozygous.
Homozygous. That side also homozygous, no? Then what is the difference between this and this?
This is dominant and this is recessive.
Homozygous dominant, homozygous recessive. So ultimately we conclude type of gametes formed here is one type. Type of gametes formed there is one type. Yes or no? आ रहा है समझ में? Are you sure? So now I'm going to write the type of gametes formed is one type. So how should I write? Type of gametes formed is one type. Type of gametes formed is one type. F1 plants. F1 plants. How will I get the F1 plants? Fertilization. By the process of fertilization, I get the F1 plants. When I write the genotype of the F1 plants, do I encircle? I don't have to encircle. Encircling is only for the gametes. Writing down inside the circle is only for gametes, not for plants of a generation. So what should be the genotype now? I will write down the dominant first. Dominant, either I write down this way. This is one chromosome, this is another chromosome. This also I can write. Or I can write down this way also. Both are correct. This way I am writing down pair of alleles. Because the situation here is this is one chromosome, this is another chromosome. R, Y, Y. So that means these plants have a pair of chromosome like this. Where this is one pair of allele, second pair of allele. So either I write this way or I write chromosome by chromosome. That way also I can write RY, RY. Either of the two methods are correct. But I prefer to write this way. I prefer to write this way. Pair of allele at a go. एक pair of allele है और ये दूसरा pair of allele है. That way it is easier also for you to remember. So let us write down now. RR, YY. What should be the phenotype of this plant?
Round and yellow.
Round and yellow. Why? What should be the shape of the seed? Round. Color should be yellow. Q?
It is strong.
Dominant. So we will write down the phenotype. Round and yellow seeds. How will I get the F2 generation now?
When
Crossing the F1. So yes, I have to cross two plants of the same type. So I will take another plant this way and cross. What is this process known as? Selfing. Selfing this is. I want to find out gametes from this plant. What type of gamete from this plant? Now, little bit of a difference I will have to remember. To find out the gametes of this plant, let me write it here. Let me write down the pair of chromosomes first. R, R, Y, Y. A pair of chromosomes. And we are now to study that these pair of chromosomes, before it shows the process of division, what happens? Or if I write this way, this is the pair of chromosomes. Before it shows the process of division, what should happen? Cell division ko concept yaad karo. A pair of chromosome before it shows the process of division, what happens? Duplication. So agar wo such hai, then I have to show something this way. That these are the two parental chromosomes and the duplicated copy is formed. Duplicated copy means alleles are same on the duplicated copy, no? So if this is R, this is also capital R. If this is Y, this is also capital Y. If this is R, this is also small. If this is small y, this is also small y. Understood? Duplication. Now, something new that you are learning is these two duplicated chromosomes, जिसको non-sister chromatids कहते हैं. You have learned this in the chapter of cell division. Non-sister chromatids. These are called non-sister chromatids. What happens to the non-sister chromatids? The non-sister chromatids will undergo the process of crossing over. याद है? Non-sister chromatids will undergo the process of crossing over and result in the formation of an X-shaped configuration. This you have studied, no? So let us see that how it happens. ये जो दो non-sister chromatids है, they always have a tendency of crossing over, something like this. Something like this. Okay? So this is capital R, this is capital Y. This is this chromosome I'm talking about. 1. What about 2 now? This is 2. So इसके alleles क्या है? Capital R and this is capital Y. तो ये हो गया strand 2. अब strand 3 and 4 let us check. This is small r and this is small y. इसमें कोई confusion नहीं है. This is small r, no? And this is small y. ठीक है? Now another concept that you're going to come across new is when the process of crossing over happens, the two non-sister chromatids they always turn, twist and separate. तो ये turn and twisting में always there is a break. और ये break होने के बाद this and this will join, this and this will join. आया समझ में? So this chromatid or chromosome they will coil and form a continuous thread. These two will join, form a continuous thread. So ultimately how many chromosomes we are getting? 1, 2, 3, 4. So now let us write down the alleles. Tell me for this first strand, capital R, capital Y. Tell me for strand number 2 now,
Capital R, small r,
and small y. Tell me for strand 4 first,
small r,
and tell me for 3.
small r, capital Y.
Is this understood? Small r, capital Y. This already in the process of cell division you had learned. When the process of duplication happens, followed by crossing over, the non-sister chromatids always they turn, twist and separate, resulting in the formation of new combinations, recombinations. So it was a recombination. Earlier, these two were together. Capital R and capital Y, these two alleles were together. Now this and this has come together. New combination has happened. No? Earlier small r and small y were together. Now small r and capital Y has come together. Recombination has happened. So ultimately, this type of parent, which is a heterozygous condition, how many type of gametes will it give rise to? Now that the cell separates, one cell will get, now that the cell gives rise to gamete, 4 gametes will be formed. One cell will get this, one cell will get this, one cell will get this, one cell will get this. 4 gametes. So this will give rise to one type of a cell, this will another type of a cell, this will result in another type of a cell, this will result in another type of a cell. So how many type of gametes? 4 types. 4 types of gametes. So let us write it down. How many types of gametes? What should I write? Capital R, capital Y. Number 1. Next, what should I write?
Capital R, small r.
Third, what should I write?
Small r, capital Y.
Fourth, what should I write?
Small r, small r.
Is this okay now? Yes. Should the type of gametes of this plant also be the same? Same. Because genotypically and phenotypically these two plants are exactly the same. Is this clear to you? Yes. आया समझ में? So the F1 plants producing how many type of gametes? 4 types of gametes. 4 types of gametes. No, the F1 plant producing 4 types of gametes. Now we want to get the F2 generation. Now we want the F2 generation. Here can we go by the crisscross method? No. क्यों नहीं?
It will be too complicated.
It will be too messy. क्योंकि 4 gametes. This parent also 4 gametes. This parent also 4 gametes. No? So in this case, the Punnett square would be very easy and convenient way of expressing. In this way, in this case, Punnett square would be very easy to express. So let us now talk about the F2 generation plants. And now we'll have to make the Punnett square. To make the Punnett square, we first have to write down the gametes. The method is exactly same like what you have done earlier. The method is exactly same like what you have done earlier. So let us write 2, 3, 4. Okay. One line is only for writing down the gametes. You have to consider one plant female, one plant male. One plant female, one plant male. So I write down the gametes. Are the gametes both ways different or same? Same. So I will start writing down the gametes. Keep telling me. ठीक है? Write the same this side. Understanding? Now, I will number them. 1, 2, 3, 4. ऐसा नहीं लिखना है एग्जाम में. 5, 6, 7, 8. 8 gametes हो गया. Now you are to write the combinations as a fertilization. 1 and 5. What should I write? 1 and 6. 1 and 7. आ रहा है समझ में? 1 and 8. आया समझ में? तो करो स्टार्ट फ्रॉम हियर। Punnett square अच्छी तरह से हाफ ऑफ द पेज में बनाना है। This boxes should be 2 lines and this should be 3 lines. ये 3 lines वाला बॉक्स होगा। चलो फटाफट। हाँ, यहाँ से लिखना है full start from here. This one is just for your understanding. ये साइड पे लिखना है तो लिख के रखो, but this you should not write in the exam. This is only for your understanding. Neatly, nicely, you have to write. Made your Punnett square. Chalo, let us complete now. So this is already done. Now I start with 2. 2 and 5. What should I write? Are you all done with this? Making on the box? Karo whatever. completing your box. It should be almost half of the page. Three lines here, then only it become neat. Otherwise it will be congested. Take care, for you it's still okay. That's too small. Done? So did you understand what I did here? Okay, let us try with this, the second line. 2 with 5, 2 with 6, 2 with 7, 2 with 8. That is how we will go. उसके बाद 3 with 5, 3 with 6, 3 with 7, 3 with 8. चलो, keep doing. Careful, you have to be check and write. If any one letter you make a mistake, later when you count the ratio, it will be all wrong. So it is not just practicing R and Y anyhow, you have to check which letter you are writing. Check which gamete you are writing. So this is the first one, 2 and 6. Check and write. 2 and 7, 2 and 8. Now 3. 3 and 5. Okay. When you write 3 and 5, see here, there's a little bit of a change. 3 and 5 you are writing, but this is the dominant, no? So we will write dominant first and then this. Dominant हमेशा पहले लिखा जाता है. If you write this and this later, that is not wrong. But there is always a way of writing also, no? That's why. So I will write this first. Now I have to take 4, 4 and 5. One and 5, 1 and 6, 1 and 7, 1 and 8. That way you go. Once you are finished with this line, then you take 2, 2, 5, 2, 6, 2, 7, 2, 8. ऐसे जाओ. Then you take 3, 3, 5, 3, 6, 3, 7, 3, 8. Then you lastly take 4. Done.
Should we also write the phenotype?
इसीलिए I told you 3 lines it should be. You will write on the genotype, then phenotype लिखने का space होना चाहिए.
Like round yellow.
Done. कोई कंफ्यूजन है? Did you understand?
This third row and sixth column.
अच्छा, अभी third and fifth जब मैंने किया, मैंने इसको पहले लिखा because this is dominant. So I wrote this first and this. I wrote this first and this. I wrote, acha, abhi, for this and this, there's nothing to worry. You can write as it is. And then this and this. Done.
This Y will be capital. No?
Which one?
Third row, second box.
Third row, second box.
No, this one.
This one. कौन सा capital होगा?
Y.
RY. Capital ही तो है.
Before that.
ये वाला.
No. This Y should be capital and the next one should be small.
I wrote this first and then I wrote this. I said no, I majorly try to start with a dominant letter. Dominant allele अगर है, start with a dominant allele. अब जैसे ये दो, now if I talk about this two, none of them are starting with a dominant allele. Then you can write as it is. But when I take this and this is starting with the dominant allele, so I choose to write this first. आया समझ में? Done? जल्दी करो. It's just like a formula that you have to apply. Which one to take with which, and then write it. Make it fast. Done? All of you done? Now let us write down its phenotype. Say suppose, take the first one. What should be the phenotype for the first? Round and yellow. Obviously, because सारे chromosomes में capital alleles ही है. Capital letters we have written. No capital letter. Capital R stands for dominant allele. Capital Y stands for dominant allele. So obviously this is round and yellow. Obviously this is round and yellow. But if I write about this, what will be the phenotype?
Round and yellow.
But this is there, no?
But it's recessive.
But it is recessive. Capital Y and small y you have to check. Can a recessive allele express in a single dose? No. It has to be two in both the chromosomes, then only a recessive allele can express. But a dominant allele one is enough. So that's why we write here also round and yellow. What about this? Round and yellow. What about this?
Round and yellow.
This is also round and yellow. What about this?
Round and green.
Round and green. So that is obvious because allele for green in homozygous condition, no? So we write round and green. What about this?
Round and yellow.
Round and yellow. What about this?
Round and green.
Round and green again. So this is round and green. What about
Round and
this?
yellow.
Round and yellow.
Round and yellow. Round and yellow.
What about this one?
Round and green.
Round and green. This is round and green. What about this?
Round and yellow. Wrinkle and yellow.
Wrinkled and yellow. Wrinkled and yellow.
Wrinkle and yellow.
Wrinkled and yellow.
Wrinkle and yellow.
And what about this last
Wrinkled
one?
and green.
Wrinkled and green. Now you tell me, look into this 16 combinations. We got 16, no? We got 16 combinations. Now in these 16 combinations, we have to now check what are the 4 types of phenotype we are getting. What are the 4 types of phenotype?
Round
We
and
got round and yellow.
Round and green.
We got round and green.
Wrinkle and yellow.
We got wrinkled and yellow.
Wrinkle and green.
And wrinkled and green. Wrinkled yellow, wrinkled green. How many round yellow we got out of 16?
9.
9 out of 16. How many round green?
3.
Check karo. 3 out of 16.
3.
Wrinkled yellow, all of you check for yourself. 3 out of 16.
1.
And this is 1 out of 16. What are we talking about? Are we talking about the different types of phenotype of the F2 plants? Are we talking about the phenotype of the F2 plants? Yes. We are talking about the phenotype of the F2 plants. Hence we write the phenotypic ratio. Phenotypic ratio is 9:3:3:1. Out of 16 plants we are getting, phenotypic expression is 9:3:3:1. So we are not talking about the genotype. Genotypic ratio is there also, but we are not talking about the genotype right now. Phenotypic ratio is 9:3:3:1 because we are talking about out of the 16 combinations. How many of them are round yellow? How many of them are round green? Wrinkled yellow, wrinkled green. So this is what we are talking about, the phenotypic ratio of a dihybrid cross. What is a dihybrid cross? The process of breeding plants in which we study two characters generation after generation. When we study two characters generation after generation, that is what is a dihybrid cross. Now, try answering these questions. Based on this, try answering these questions. करो, keep doing, फटाफट. Based on this, I am giving you will get a typical question in your paper that when round and yellow plants are crossed with wrinkled and green plants, the plants obtained in the F2 generation are such and such number. They will just give you a number that is just to confuse. The number has nothing to do with your answer, remember? Now the rest of the questions. Understood what I told you? So you will have typically, maybe in the next class again I can give you a question for revision. When round and yellow plants are crossed with wrinkled and green plants, the number of plants obtained in the F2 generation are something 550 plants are obtained in the F2 generation. What is the phenotype of the F1 plants? Like that. How many plants? Now, how many plants means you have to take out of 16 only now? How many plants show parental phenotypes? How many plants are recombinants? Okay, first try to write down the answers for the first 4 questions. The fifth, that is the statement of the law I'll give you. I'll write down here. Yes. I'm giving you the answer for only this. The rest I'm going to ask you in 2 minutes. फटाफट करो. क्या हो रहा है? Second, how many plants show parental phenotypes?
10. Because the required also.
आया सो हम समझ में? नहीं. Parents will consider this. Okay. So how many plants are this you check, and how many plants are this you check. These are the parental combination, parental phenotypes. ये नहीं लेना है. This is F1 plants. Now, you write down first, then I'll explain you what is the meaning of this law. फटाफट करो. We don't have too much of time. हाँ?
Recombinance is new.
New combination जो crossing over से मिला है. आया समझ में? Doubt है? हाँ बोलो.
6 by 16.
6 by 16 क्या?
How many plants are there?
How many plants are recombinant? Very good. 6 by 16. These are the recombinant plants, no? These are the recombinant plants.
So 10 by for the second one it is 10 by 16.
10 by 16.
And the second is
New combinations जो बना है, these are recombinants, no?
In answer, we can write only 6 plants.
6 plants. आ रहा है समझ में? Confusion है तो पूछो. This concept should be clear because this is just the basic thing. Why 10 by 16? My question is, how many plants show parental phenotypes? अभी parental phenotypes. Which are the parental phenotypes? Round yellow, wrinkle green. So how many plants are round yellow? How many plants are wrinkle green? Now this is the basic thing. जरूरी नहीं है कि in your paper they will ask you the same pair of traits and to do the same type of cross. They will give you an application question where this may or may not remain same, the pair of traits. And as I said, they will give you questions on that after breeding such and such type of plants, some 1200 plants are obtained in the F1 generation. What is the proportion of plants that you get as parental phenotypes? What is the proportion of plants that you get as recombinants? So whatever number, I'll give you one type of a question next time from the paper. So based on the number, you can easily do it. It's no big calculation that you have to do. Only this concept to remember. So that's why I gave you this. Always these are parental and these are new combinations. Once you are clear about how to do it and what are these proportions and the ratio, and if it is a dihybrid cross, the ratio has to be this. There is no other alternative. 9:3:3:1 only. There are alternatives, but that is not there in your syllabus. You are learning only one concept. आया समझ में? And this is the statement. पहले statement लिखो, then I'll explain it. फटाफट लिखो. Statement of the law. What are we studying? Law of independent assortment. Independent मतलब
Free.
अपने आप. Assortment मतलब assortment मतलब arrangement. Okay, now let us see the law. What does the law say? When two parents with two or more contrasting traits are crossed, when two parental clans, no? Two contrasting traits, no? Here we are saying 1, 1, 2, 2. We are considering two pairs of contrasting traits. So the law goes this way. When two parents with two or more, it can be more also. Dihybrid, polyhybrid भी होता है. फिलहाल you are only studying dihybrid. When two parents with two or more contrasting traits are crossed, the inheritance of one pair of trait is independent of the other pairs. Meaning what? The coming together of these two alleles or these two traits in one gamete, is it dependent on 2, 3 and 4? Or is it dependent on the second and the third and the fourth chromosome? No. The first chromosome will as it is come and form or become a component of a cell. So the coming together of capital R and capital Y together to form a gamete is independent of the other pairs. The coming together of capital R and small y to form this gamete is independent of 1, 3 and 4. It is not dependent on these pairs at all. Coming together of this and this to form this gamete is independent of this and this pair. Are you understanding what I'm trying to tell you? So this concept says that it is not that for coming up together of this and this also should come in the same. No. Every single chromosome has two alleles. One for color, one for shape. Every single chromosome has two alleles. One for shape, one for color. One for shape, one for color. One for shape, one for color. And the coming together of these two alleles, meaning two traits, no? This determines roundness. This determines yellowness. This determines wrinkled. This determines yellow. So every chromosome has got one allele for shape, one allele for color. And coming together of these two alleles to form a gamete is independent of the other pairs. Independently, the alleles are arranging to form gametes. Independent assortment. Understood? Independent assortment. Why? The law is termed as independent assortment because in the F1 generation, this is a concept that has been ultimately understood. Every gamete has a pair of allele which is independently assorted in the form of a chromosome. So this is the statement that you have to understand and remember. Aaya samajh mein? So with this we actually come to the end of this chapter. You can very well prepare for your exams, but the next class will solve some questions also. For the time being for your exam, you just go through the text. You will be able to solve questions, not a problem. But for the long run, to complete the chapter with questions, the next class will actually solve some few more questions from the text. And if possible, the next day, maybe we'll introduce the next chapter. The next chapter that I'm going to take up would be photosynthesis. Okay. So either, if next class is possible, good. If not, then the next class I'll start with the next chapter. Anything to clarify? Any doubts, confusion? Anything from here or previous? Nothing? Okay, we complete. All of you are done with this. So go through the text questions for your exam. You may just go through some of the text questions. This is a different book. Okay, now in this case, just one more minute. Name the following. Name the following. Keep answering. From your book only I'm taking. For Selena, you will just try answering. The genetic makeup of an organism. Genotype. The unit of heredity is genes. A hereditary disease in which the blood does not clot. Hemophilia. Check the spellings also. The type of gene which is in the presence of contrasting trait not expressed. Recessive. The physical expression of genes. Phenotype. A nitrogenous base in the DNA. A nitrogenous base in the DNA. Any of the following because they have not mentioned anything. A cross between two parents having one pair of contrasting traits. Monohybrid. The repeating component of each DNA strand, lengthwise. Lengthwise. I repeat. The repeating component of each DNA strand lengthwise. Nucleotide. Repeating component of each nucleotide lengthwise. They are not talking about the component of a nucleotide. If component of a nucleotide, then the answer is different. A pair of corresponding chromosome of the same shape and size and derived from each parent, homologous. So there are questions from here also. You may go through. Selena also has got a lot of questions. So as it is, go through the question, prepare the test as of now, and we'll try solving, going through the questions again in the next class. Okay. Thank you.